Today I'm going to be explaining mitosis versus meiosis, and on the left side you see mitosis, and on the right side you see meiosis. So first things first, they are both cell division processes. The one thing that differs is their purpose. Mitosis is for growth and repair, while meiosis is for sexual reproduction. But mitosis can also be used for reproduction, except it has to be asexual reproduction because, as you can see, the result is identical cells, while in meiosis it is non-identical or genetically variable cells. So moving on to the labeled version of these biology cookies, we both see interface is very similar, except in meiosis you have chromosomes from both parents. Prophase, in mitosis prophase, the sister chromatids align to form a chromosome, and the early mitotic spindle starts forming. And in prophase of meiosis, the homologous pairs line up together and exchange information at the chiasmata, and this is called crossing over. So you see the parent 1 and parent 2's information are crossing over, so this is where the genetic variation comes in. In metaphase, they both line up at the metaphase plate, so that's something similar. Another thing that's similar is the kitinochore microtubules pulling towards the centrosome, while the non-kitinochore microtubules extending towards each other. So here you see the same thing, except the recombinant chromosomes are lined up with their homologous pair. And in anaphase, they both split apart, except this one you have two sister chromatids splitting up to form their own chromosome, while over here you see a full chromosome with two sister chromatids still attached to each other, splitting apart from their homologous chromosome pair. And in telophase, we see there is a cell division at the cleavage furrow, and cytokinesis actually divides it into two cells. Mitosis is done. So here we see two 2N or diploid daughter cells, which are identical, so there's no genetic variation, it's just a clone. But in meiosis, they are genetically variant, and the process is still not done. In prophase 2, we see something similar to prophase 1. The early mitotic spindle starts forming, and the sister chromatids are still aligned, and then in metaphase, they line up at the metaphase plate, and the mitotic spindles start pulling. Same kitinochore and non-kitinochore microtubules work to pull the sister chromatids apart. In anaphase, the sister chromatids are getting pulled apart to form their own daughter chromosomes. So you can see the sister chromatids are separating. And then in telophase, it's similar to telophase 1 of mitosis. So they split at the cleavage furrow, and instead of 2 cells, you have 4 haploid cells. So they have half the chromosomes that were originally there in the cell. So there we have it, mitosis versus meiosis.